Dear colleagues, my name is Marco Weiergräber. It's my distinct honor and privilege to welcome you as the founding editor and editor-in-chief of Translational Brain Rhythmicity, an online peer-reviewed open access journal. In my capacity as editor-in-chief and on behalf of the editorial board members, I would like to forward a very warm welcome to you, the readership of our new journal. Neuroscience is an extraordinary, fascinating field composed of numerous subdisciplines. However, none of these subdisciplines remain isolated. Indeed, they all share to a specific extent common aspects and are to be investigated considering their functional interdependence. Translational brain rhythmicity covers the functional integration of these subfields and their impact on brain rhythmicity. Research within the last decades has gained tremendous insight into the mechanisms of proper brain function, but also pathophysiological aberrations responsible for neurological and neuropsychiatric diseases. Many of these important findings are related to alterations in biochemical pathways, signal transduction cascades, and ion channel dysfunction, suggesting that we already know a lot, but clearly not all, about the molecular and cellular background of neurological and neuropsychiatric diseases and epileptic disorders. In drug research and development, most novel pharmacotherapeutic approaches and recent interventions are based on molecular and cellular findings in neurons, and in many cases these novel approaches have failed to exhibit efficacy or effectiveness and positive benefit-risk ratio in patients following independent scientific assessment of clinical trials. A very prominent example for this development is Alzheimer's disease, in which most phase 3 clinical trials failed within the last 10 to 15 years, although many candidates were novel, first-in-class drugs, and highly promising due to basic and preclinical research results. This observation clearly tells us that there is much more we have to learn, and we are still lacking sufficient information about the higher structural and functional organization of the central nervous system. It feels mandatory to us to overcome the gap between basic neuroscience and systemic neuroscience and to contribute to the overall understanding of how the brain and its subsystems actually work together and interact. The journal Translational Brain Rhythmicity is thus dedicated to help unravel the functional interdependence between molecular and cellular neural processes and their impact on neural ensemble activity, that is, the physiology and pathophysiology of neural circuitries and networks. Importantly, studies presented in Translational Brain Rhythmicity are not restricted to the mammalian or human brain, but they can also include CNS rhythmicity studies on brains and nervous systems and other taxa, as long as they contribute to the overall understanding of the highly differentiated CNS structures, neural circuitry rhythmicity, and oscillations. Our journal is specifically dedicated to translation of molecular, cellular, and systemic neural experimental approaches from animal models to human neurological and neuropsychiatric diseases. Methodologically, the journal is not only limited to cellular and systemic electrophysiological in vitro and in vivo techniques, but also to molecular, genetic, and behavioral approaches that affect neural activity on the cellular and thus also on the circuitry and network level. We also welcome submission of clinical trial results of CNS drugs that affect or exhibit modulation of brain dysrhythmia. As a prominent forum of scientific exchange, Translational Brain Rhythmicity encourages submission of different manuscripts formats, for example, full-length original articles, short research letters, review articles, methodological manuscripts, case reports, clinical trial outcome reports, best practice guides, commentaries, controversies, congress reports, and finally, letters to the editor. The editorial board of Translational Brain Rhythmicity ensures application of highest scientific standards, conforming to strict international peer reviewing processes and editorial standards of the scientific community. Translational Brain Rhythmicity is published 6 times a year. In order to spread scientific information fast, the reviewing process is aimed to last not longer than 2 months. In order to disseminate outstanding manuscripts and their findings rapidly, a fast-track reviewing system is installed that guarantees publication within 2 weeks. This approach enables translational brain rhythmicity to timely react on fast new developments in the field. If you would like to push a specific topic in translational brain rhythmicity, we strongly encourage you to submit proposals for specific issues in which you can serve as a guest editor. Besides, the editorial board of Translational Brain Rhythmicity will periodically announce calls for specific issues on current hot topics. Finally, I strongly encourage you to submit your valuable research through Translational Brain Rhythmicity. We hope that you will find Translational Brain Rhythmicity a useful and informative outlet for your research and a valuable source of information, evidence, and critical debate. We hope to hear from you soon and highly appreciate your feedback. Best wishes and take care.